Good morning, children. Today let's study about the structure of nucleus. As we have learned in the last chapter, nucleus was discovered by Robert Brown. Inside the nucleus, there are many thread-like structures which are called as chromatin. The chromatin is mainly composed of DNA and proteins. We learned this part towards the end. The name chromatin was given by Fleming. Let's study in detail the structure of nucleus. The structure of nucleus, it includes the nuclear membrane, nucleoplasm, chromosomes and nucleolus. The four parts, children: nuclear membrane, nucleoplasm, chromosomes and nucleolus. The nuclear membrane. The nucleus is covered by a double layered membrane. Here in the picture you can clearly see the nucleus is having a covering, or the nucleus is covered by a double layered membrane, the outer membrane and inner membrane. The outer membrane is connected to endoplasmic reticulum, and it's actually the rough endoplasmic reticulum. You can see the ribosomes are attached on it, or we can say the outer membrane is in continuation with the endoplasmic reticulum. Here you can see the inner membrane. The inner membrane encloses here the outer membrane and the inner membrane. Between outer and inner membrane, the space, it is called as perinuclear space. So one more time, children, the nucleus is covered by a nuclear membrane, the outer and inner membrane. In between these two membranes, the space is called as perinuclear space. And when you look at the picture, here we can see, children, this nuclear membrane, it's not continuous. In between you can see there are some gaps are there, or pores are there, which are called as nuclear pores. Now just have a look at the picture. We can see this is the outer membrane, this is the inner membrane. These outer and inner membrane gets fused at certain regions, and the fusion of these two membranes, it actually forms the nuclear pores. And it is through this gap, or through these nuclear pores, the movement of RNA and proteins takes place in and out of the nucleus, from the nucleus to the cytoplasm, and from the cytoplasm back to the nucleus. The movement of RNA and proteins, it takes place through these nuclear pores. Now, in the case of eukaryotes, in certain cases, we can see at maturity the cell lack nucleus, or at maturity the nucleus is absent. Examples: in the case of mammals, in the case of human beings, in the case of mammals, the erythrocytes or the RBC, erythrocytes means RBC, they lack nucleus. And in the case of plants, among vascular plants means which have the vascular tissue, xylem and phloem. We have learned the structure of xylem and phloem, and in the case of phloem, it consists of seed tubes, companion cells, phloem parenchyma and phloem fibers. In the case of seed tube, it is made up of seed tube cells. These cells of the seed tube at maturity, they lack nucleus. So two examples we have learned, children. Among animals, we have learned in the case of mammals, in many mammals, at erythrocytes or RBC, they lack nucleus. Among vascular plants, the phloem, which consists of the seed tubes, in the case of seed tubes, at maturity, the nucleus is absent. Now, we have learned about the nuclear membrane. The next part we can see the matrix inside the nucleus. The nuclear matrix, it is called as nucleoplasm. Now, this nucleoplasm, it consists of a spherical structure which is called as nucleolus. It consists of thread-like structures which are called as chromatin. Now, this spherical structure which is present inside the nucleoplasm, it is called as nucleoli, or it's a plural word, or nucleolus. This nucleolus is not a membrane bound structure. It is not covered by a membrane. And its main function is a site of ribosomal RNA synthesis. Children, last time just revise fast the structure of nucleus. The nucleus is covered by nuclear membrane. The nuclear membrane is double layered, outer and inner membrane. The outer membrane is in continuation with endoplasmic reticulum. And in between the two membranes there is a space which is called as perinuclear space. And in between here we can see there are some gap, or the nuclear pores are present through which the movement of the proteins and RNA take place. Inside the nucleus, the matrix is called as nucleoplasm. The nucleoplasm consists of the spherical structures which are called as nucleolus, and the thread-like structures which are called as chromatin. Now, next chapter we will learn about the cell division. And when we consider the life cycle or the cell cycle, the life cycle of a cell, or what we call it as the cell cycle, we can see in the life of one cell, there are two phases, a dividing phase and a non-dividing phase. That means when we consider the cells, the cell, it undergoes division, and the cell at a time it's not undergoing division. The non-dividing phase, when it is not dividing, that phase it is called as interphase. And during interphase, the structure of nucleus is very clear. Children, we will learn later. During cell division, in different stages of cell division, the nuclear membrane and nucleolus will disappear. So, if you want to study the proper structure of a nucleus, it is during the interphase. So, during interphase, the nucleus consists of nuclear membrane, nucleolus, and it consists of a loose and indistinct network of fibers which are called as chromatin. So, chromatin is actually a network of fibers which are present in the nucleoplasm during the interphase. Interphase means when the cell is not undergoing division. This chromatin, it actually consists of DNA and some basic proteins which are called as histones. It also consists of some non-histone proteins and RNA. Now, at the time of cell division, these chromatin threads, it undergo condensation and it forms the structures what is called as chromosomes. Here in the picture we can see, children, this part which is written as A, T, G, C and all, it is DNA. The structure of DNA we will be learning in another chapter. So, this double helical structure, this blue colored double helical structure, it is DNA. DNA along with the proteins, histones, it forms what is called as chromatin. This chromatin, it undergo coiling, it undergo condensation, and it forms the structure what is called as chromosome. The same picture one more time, children. Here this purple color, this part, it is the DNA. DNA along with the histones, it forms the chromatin. These chromatin threads undergo coiling and condensation and it forms the structure chromosome. Yes, children, now let's study the structure of chromosome. Each chromosome, it consists of two chromatids. Here we can see, children, this is a chromosome. Each chromosome, this is one chromatid, and here this is the other chromatids. These two chromatids together, it forms a chromosome. And for each chromosome, it has a constriction which is called as centromere or the primary constriction. Every chromosome has a primary constriction and the primary constriction is called as centromere. These two chromatids are held together by means of centromere. The two chromatids of the same chromosome, it is called as sister chromatids. Now on here in this picture we can see this part, it shows centromere. On either side of the centromere you can see a pink color or a disc-like structure which is called as kinetochore. So the primary constriction is called as centromere. On either side of the centromere, the disc-like structures are called as kinetochore. At the time of cell division, the spindle fibers, it gets attached on kinetochore. That part we'll be studying in the next chapter. Only this much now we have to learn. The chromosome, it has a constriction which is called as centromere. On either side of centromere, the disc-like structure, it is called as kinetochore. Now, based on the position of centromere, the chromosomes are classified mainly into 4 different types: metacentric, submetacentric, acrocentric, and telocentric. Now, the first one here is metacentric. In the case of metacentric chromosome, the centromere is in the middle. This gray color part is the centromere. In the case of metacentric chromosome, the centromere is in the center, so that it has 2 equal arms. When the centromere comes in the center, the 2 arms are equal. This type of chromosome is called as metacentric. In the case of submetacentric chromosome, the centromere is slightly away from the middle. In the case of metacentric, it is in the center. Here in the case of submetacentric, it is slightly away from the middle, so that one arm is longer, the other arm it is shorter. It is called as submetacentric. The third one here, in the case of acrocentric chromosome, the centromere is situated close to one end. Now what happens here, children? It forms one arm is extremely short and the other one is very long. It is called as acrocentric. Now the last one, in the case of telocentric, the centromere is in the terminal. It is at the end. Children, this diagram you have to learn for the exam. Now, here we have learned, children. Just have a look at the picture. We can see this is the centromere or the primary constriction. Other than the primary constriction, if it is having any other constricted or narrow region, it is having secondary constriction. A narrow region or a constriction at any point on the chromosome other than the centromere is the primary constriction. Other than the centromere, if there is any constriction, it is called as secondary constriction. Now, when the secondary constriction is over here, a part you can see what is called as satellite or chromosomal satellite. It is the part of the chromosome which is separated by the secondary constriction. Now the last part here we can see, children, in the case of this eukaryotic cell, there are certain membrane bound minute vesicles are present which are called as microbodies. These microbodies contain various enzymes. It is present in both plants and animals.